Hi, so this is chapter 27, the reproductive system. We're going to look at the overview and begin with the male reproductive system first. Of course, if we take a look at this first slide, the reproductive system is to produce gametes, which are haploid cells, so that we can have basically sexual reproduction, right, to produce offspring that are different, genetically different than the parents. And so the process that creates gametes is called meiosis. So we're going to go over the meiosis that happens in the male gonad, which is the testes, and the female gonad, which are the ovaries. So the word gonad refers to organs that produce these gametes. And if you remember the word haploid, gametes are haploid. They only have half the normal number of chromosomes. So if you think about that number, right, how many chromosomes do humans have? We have 23 pairs of chromosomes. So we have 46 individual chromosomes in every single one of our cells. Each chromosome has a pair. But when you create a gamete through the process of meiosis, you are having, right, making in half the number of chromosomes. So each gamete has 23 individual chromosomes. Now I want to review, and you probably went over this in Bio 3, that the letter N refers to the number of gametes. I'm sorry, the letter N refers to the number of chromosomes, the different chromosomes that you have in a cell. So we have 23 different chromosomes. Chromosome 1 is different than chromosome 2, is different than chromosome 3, etc. So N for humans is 23. So a haploid cell is referred to as an N cell. The letter N is going to stand for haploid. And then a diploid cell with the pair of chromosomes will be 2N. Okay, so that should ring a bell from Bio 3. So we'll look at meiosis in the male and meiosis in the female pretty soon. Okay, so meiosis also involves genetic recombination. So there is built into the process of meiosis a shuffling and mixing of the genes so that every single gamete that you make in your life is going to be genetically different than another gamete. So every single egg a woman produces in her life is genetically unique. Every single sperm that a male produces in his life is genetically unique. All right, so let's look at the second and third and fourth overview points. The reproductive tract is going to be a tract, just that. It's going to be a path that is going to receive, it stores, and it transports the gametes. Accessory glands, just like the digestive system had accessory glands, right? The liver or pancreas are accessory organs and the salivary glands. The accessory glands of the reproductive system is going to secrete into the ducts or into the tract to help those gametes survive or provide nourishment for the gametes. Then there's going to be the perineal structures, the external genitalia we'll go into. All right, so if you want to just take a look at this picture, we have a nice big ovum. Okay, so ova is plural, ovum is singular for an egg. And then the sperm, you can see all the different sperm on top of the egg in the process of trying to fertilize that egg. All right, so let's start with letter number 2, the male reproductive system. So we're going to begin with the testes. So the testes are the gonads, again, of the male reproductive system, and this is where your sperm are created. So it produces sperm, but also it has a role in producing male androgenic hormones. So androgenic hormones are basically saying male hormones. The most prevalent and well-known of all the androgens is testosterone, and there's going to be smaller, like lesser prevalent hormones that are testosterone-like, but testosterone is an androgen. All right, so if you look at the scrotum here, right, they descend into scrotal sacs during the last 2 months of development. And why is this? So I asked the class this, why is it that the testes are outside of the abdominal cavity, or they're not inside the pelvic cavity? And the reason is the temperature of the abdominal cavity is too warm for appropriate sperm development. So the testicles have evolved to be outside of the abdominal pelvic cavity, and so they're housed in the scrotal sacs or the scrotum, right outside the body wall. So it's all about the perfect temperature for sperm development. If it's too warm, the sperm are harmed, and there's mutations and deformities that can happen to each sperm cell. So the scrotum has a couple of muscles that really mediates the temperature of the testicles. So letter A, the cremaster muscle. This is skeletal muscle. So if you want to take a look at the picture here, so we have the layers of the scrotum, we have fascia, then we have a muscle called the dartos muscle. And then if you look here, we have the cremaster muscle. So the cremaster muscle is actually a very robust muscle, and it's an extension of your internal oblique. So you know that the oblique muscles are in the abdomen, but there's an opening here. So this is coming back again to our inguinal ligament, right? We talked about this in the middle of the semester. So this inguinal ligament has two rings, remember, two openings. So this is our superficial ring that faces the skin, and the deeper ring on the inside. So the internal oblique muscle continues on and comes through the superficial ring and completely envelops the testicle. So this is the cremaster muscle. This, when it contracts, it raises the testicles up closer to the abdomen, and when the muscle's relaxed, it lowers the testicles away from the abdomen. So this is all about temperature control. So if a man is cold, right, this muscle is going to contract and raise the testicles closer to the body so it can be warmed. And if the man is very warm, right, then the muscle will relax and allow the testicles to move away from the abdominal cavity to dissipate heat. The dartos muscle is the same concept, but it's a smaller muscle that's just in the skin of the scrotum. So you see layers of the scrotum. So this is basically skin. Okay, it's skin, but then right underneath the skin you have this muscle. I'm going to show you this other picture here where we see this nice robust pink, right? We recognize that color now for muscle. This is cremaster. So cremaster is very big and robust. And if you look at the skin, so there's your outer layer of the skin, which we see here, then right underneath that we're going to have a little, it's drawn in just very light strands of pink. So this is the smooth muscle of the dartos right underneath the skin. So what is dartos doing? Dartos is going to either wrinkle the skin when it's contracted, or when it's relaxed, it allows for the scrotum to have a smooth skin. When the skin wrinkles, it holds in heat. When the skin is relaxed and smooth, it can dissipate more heat. So it's a mechanism that regulates, the micro fine tunes the temperature. And I have kind of a funny image here I want to show you. The ways to lower your sperm count. So this is real, and if you later in life want to have children and maybe can't, like hopefully this doesn't happen to you, but I personally have some friends that have waited for a while to have children, and then they found that when they're later in life, it's more difficult to have children, and then you start worrying about sperm count. So being in a very warm environment can harm the sperm and lower your sperm count. So being in a hot tub, for example, is very bad if you want to have children. Wearing tight pants, people will actually say, fertility specialists will say not to wear tight pants, or the kind of underwear that holds, I was going to say called tighty-whities, but which is right here, but they hold the scrotum and the testicles, up close against the body, and so it's just a warmer environment that's needed for sperm development. So these are all no-nos if you want a healthier sperm count. Okay, let's go back to number 2. So let's go back to our picture here. So let's stay here and take a look at our next term on the list, number 2, the Tunica Albuginea. So the Tunica Albuginea is basically if you think of it as like a capsule on top of, a lymph node, it's that same kind of functionality. It's connective tissue capsule that is on top of the testicle, right? So it gives the testicle some rigidity and some structure. because the internal tissue is very soft and delicate. And this Tunica Albuginea is going to form walls. It's going to sort of form those little segments inside of the testicle. So those walls are called septa, right? Just like the nasal septum is a wall between the two nasal cavities. These are the septa that are created inside the testicle. Inside, we can see that the testicle has a coiled, tons and tons of coiled structure called seminiferous tubules. These seminiferous tubules are where the sperm are made. So this is where meiosis occurs, and we're going to take a look at this. Okay, so the lobules, these individual units here, these are the lobules of the testicle. They are defined by the septa. All right, so let's take a look at the next portion of the outline, number 3, the seminiferous tubules. So again, that's your purple coiled structures here that basically is 80% of what the testicle is. And this is where spermatogenesis occurs. So spermatogenesis is the whole process of making sperm. So let's take a look at the next slide here. This is spermatogenesis. So when you start off with forming sperm, you always have a population of stem cells in your gonads for man and women. For a male, the stem cells are called spermatogonium. Okay, spermatogonium are diploid. This is a stem cell. So stem cells can reproduce themselves. They can divide and create another stem cell to sort of keep a constant population of stem cells in the testicle, or they can go on and undergo meiosis. So when they go on and undergo meiosis, the first step is to produce a primary spermatocyte. Then the primary spermatocyte is going to form a, here's the primary spermatocyte, we'll start here, right? It's going to form, after meiosis 1, a secondary spermatocyte. After meiosis 2, then we have spermatids, okay? So the trend is we have the spermatogonium, the stem cell, differentiating into a cell that will undergo meiosis called this primary spermatocyte. And after meiosis 1, so remember meiosis has 2 phases, right? There's meiosis 1 and there's meiosis 2. After meiosis 1, you have your secondary spermatocytes, excuse me. And after meiosis 2, meiosis is done and you have your finished gametes here. These are your spermatids. So the spermatids are haploid, okay? And these are basically genetically done. They are genetically different from each other. They're haploid. The only thing that needs to happen now is that they need to change from a round cell into what we can recognize as a sperm cell. In order to do this, there's a second process called spermogenesis. Okay, so if you look at your outline, I have the word spermatids will undergo spermogenesis with the help of nurse cells, are also called Sertoli cells, and I'll explain that in just a second. So spermogenesis is the process of losing all this cytoplasm and all the organelles is going to weigh the sperm down, because the sperm, otherwise known as spermatozoa, are streamlined cells that basically just are delivery machines. They just want to take the DNA from the male and deliver it to the ovum. So they have to be able to swim up the female reproductive tract. And so all that baggage and extra cytoplasm and organelles that they don't need, they're going to shed that, and they're going to actually also grow a flagellum, right? So here is what it looks like inside of the male reproductive system, the seminiferous tubules. The spermatogonium are going to be on the edge or on the periphery of that seminiferous tubule. So it's a tube, right? We're looking at just a little cross section of this tube. The tube is hollow. Here's the lumen. And then we have here's that word, spermatogonium. Our stem cells are going to stick on the periphery, right? They're going to line the wall of our seminiferous tubules. As they undergo meiosis 1 and meiosis 2, they're going to travel towards the lumen. So let's see if we can find our primary spermatocyte. This is here. Okay, so it's the first one formed from our stem cell, so it's going to be closest to the stem cell. Then as it divides, Again, in meiosis 1, it's going to be a secondary spermatocyte. And then the secondary spermatocytes will undergo meiosis 2, and they're going to form spermatids. So here's spermatids, right? So spermatids are going to be closest to the lumen. And then the spermatids will undergo spermogenesis to change morphology, right? So that word morphology means shape, into the sperm. So meiosis takes care of the genetic, having the chromosome number and also creating diversity. And then spermogenesis takes care of the morphology change. So at the lumen of a seminiferous tubule, you're going to find your spermatozoa, right? So the spermatozoa, this is showing you that they're completing spermogenesis, so they're becoming spermatozoa here. Now I want to show you a different population of cells. Besides the cells undergoing meiosis, there are things called nurse cells or Sertoli cells. These cells are going to help the sperm move physically from the periphery of this seminiferous tubule towards the lumen. And during spermogenesis, when you're shedding all the cytoplasm, all that waste, the cellular debris, is taken in by those nurse cells. So they're going to act as phagocytes and take up that debris. All right. So let's actually go back before we look at the histology slide. So for every one spermatogonium, you get 4 gametes, right? 4 haploid spermatozoa. Just keep that in mind. I think I'm done here. So let's look at the histology slide. So this is a histology slide that you're going to be responsible for knowing the seminiferous tubules of the testes on the final exam. And what you see here is the testicle, right? So this is a testicle with a cross-section through, and every single round unit is a slice or cross-section of the seminiferous tubule. So along the periphery, again, these cells, the very first cells you see along the periphery, those are going to be spermatogonium, the stem cells. And as you move towards the center, they're undergoing meiosis. And then in here, near the lumen, you're going to have your spermatozoa. Sometimes the tails of the spermatozoa, the flagellum, will come into the lumen and make the lumen look like it's filled with something, okay? So I want to show you the next thing here, and then I'll show you a blow up. So now, there are also cells outside the seminiferous tubule, right? What are these cells? These cells are called interstitial cells of Leydig, which is number 4 on your outline. And these are the cells that are endocrine cells. They are going to secrete the androgen hormones. Okay, so basically testosterone. They're going to secrete testosterone. All right, so let's take a look at a closer look. So this is our seminiferous tubule. So here's what I want you to know for the final exam. You're going to want to recognize this, right? You're going to see something that looks like this. You're going to say that is a slide of testes. That is a bunch of seminiferous tubule cross sections. So one thing you want to know and be able to write out is seminiferous tubule. You also want to be able to point out interstitial cells of Leydig, right? So you want to be able to write that out as well and know that they secrete androgen hormones. When you get a closer look, there are only 2 populations of cells that I require you to know, and that is the ones at the periphery. So because these are nice and easy, right? I know that if they're right back here, that this is our spermatogonium. And then I can see very clearly the different shaped heads of our spermatozoa. So this is something that you should be able to recognize too, which I think is fairly easy. So the only 2 cells that I want you guys to pick out are the spermatogonium and the spermatozoa. All the different ones here are too sort of difficult, not difficult, but just it's not something that I require that you do. Okay, so that's it. All right. Now, let's talk about the anatomy of a sperm. So if you look at your outline on number 3, under the word spermatozoa, there's a head, an acrosome, a middle piece, and a tail. So here is our spermatid. It's undergoing spermogenesis, right? It's shedding a lot of cytoplasm. It's losing organelles it does not need. Basically, the only 2 organelles that a sperm has will be the nucleus, which has the DNA, of course. And then we're going to retain the mitochondria, and the mitochondria are going to line up here in what's going to be called the middle piece. Not too exciting. The middle piece. So the head of so here's the head. The head of the sperm is going to contain the nucleus and something called an acrosomal cap. The acrosomal cap is going to be a specialized structure filled with enzymes that are going to be able to eat and digest the covering on top of the ovum as it's penetrating and trying to get into the plasma membrane and enter into the ovum. So the acrosomal cap is going to be filled with enzymes that are going to digest the surface of the egg. Okay, so there's a couple layers on top of the egg that we'll get to when we talk about the female reproductive system, but that acrosome is going to help digest the way through. The mitochondria in the middle piece is very important because those are going to provide ATP in order to move the flagellum, right? So the flagellum portion is known as the tail, and it's a long, basically just microtubules arranged in a way, but it's going to help propel the sperm forward, right, up the female reproductive tract. So this is a really cool sperm motility segment that I will put on your module. So if you want to pause here and take a look at the sperm motility clip, I would think that would be good. And then you can come back and finish the lecture and part 2 of the lecture.